Here we're looking at another piece of liver. This isn't a human liver. This comes from some sort of animal. The species is undetermined. And this animal has been injected with carbon particles, and the purpose of injecting the animal with carbon particles is so that the Kupffer cells, the macrophages which are present in the sinusoids in the liver, will take up these carbon particles, and this in turn, because carbon is very visible in the microscope, will make individual Kupffer cells visible. As we look at low magnification here, what we can see are largely these are central venules here, and they're surrounded by radiating plates of hepatocytes, which you can see here. Curiously enough, if you look at low magnification like this, you can actually detect areas where cells stain more darkly. Now this isn't just for carbon particles, areas like this, and forming kind of anastomosing plates like this, and in fact these represent zone 1 hepatocytes, the ones which are closest to the portal canals and furthest from the central venules. And you'll see that in general the radiating plates of hepatocytes closest to the central venules tend to stain a more pale color, and this is because they have less organelles and store less glycogen in particular than do those cells closer to the portal canals. We can see structures like this, which are very large feeder branches, which will give rise to the smaller vessels at the portal canals, and then some very large vessels like this is probably a hepatic vein. So if we go up in magnification now, and if we look here, we have a portal canal structure here. So let's see if we can identify the elements of the portal canal. This large thing here is going to be a portal venule here. Here we have a very large bile ductule verging on the size of one of the hepatic ducts. In fact, the epithelium in this case appears to be stratified on this side here and simple on this side here, and most likely it's still simple. It's just the plane of section. Where, you might ask, is the hepatic arteriole? The arterioles generally are very small, and the hepatic arteriole in this instance is just right here, and we can see endothelial cells and one layer of smooth muscle cells surrounding. It's actually found in close association with this bile ductule. We can also see some lymphatic vessels. There's one here, there's one here, and there's one here, and we can see the connective tissue, the pink staining material, which wraps around all of these structures and which is contained within this space called the space of Moll. And then for the sharp-eyed among you, you'll notice that there are actually very small bile ductule here and a very small bile ductule here. These are in fact probably, given on their location, canals of Hering. Now, the important thing from this particular slide to see is we can see the radiating plates of hepatocytes radiating outward, and we're going to increase in magnification just a little bit more so we can see the feature that this slide is particularly designed to show us, and that is the Kupffer cells, which have phagocytosed and taken up the carbon particles which were injected into this animal, so they've been taken up from the blood. And so the cells that are filled with these carbon particles appear very, very dark stained, very black. We can see them here and here. And particularly what we're looking at is that there's virtually no carbon taken up by the hepatocytes themselves, which you can see here. And as we move closer toward a central venule, so here we have the nucleus of an endothelial cell which is lining a sinusoid. And here we can see carbon particles accumulated in a cell, which, as we can see quite clearly here, is actually in the sinusoid, not in the hepatocytes. Here again, a cell in a sinusoid, in a sinusoid, in a sinusoid, and these are the Kupffer cells, the tissue-resident macrophages that are found in the sinusoids of the liver, and which play very important roles both in heme metabolism, in iron metabolism, and in secreting inflammatory cytokines in times of liver damage, which in turn stimulates other cells to secrete connective tissue and can cause cirrhosis. Finally, to complete our look at liver, let's take a look at a liver that's been stained with a special silver stain, which is specially designed to demonstrate reticular fibers. And reticular fibers are the special type of collagen type 3 fibers that form a parenchymal framework in many organs, the liver included. So we can see nothing really at the lower magnification other than some portal canals or larger structures that feed portal canals, such as this one here, and perhaps some very large central venules, or these may in fact be portal venules. If we increase the magnification a bit and we have a look here, we can have a look at this structure here in a second, which is a portal canal. But really what we want to be able to see here is the dark black staining material. This is the stuff that's been stained with silver. And this black staining material are very delicate fibers that form the supporting meshwork for the sinusoids. And effectively the reticular fibers in and around the sinusoids are occupying the space of DISA between the endothelial cells and the hepatocytes. So if we look at a little higher magnification again, then and take a look at the portal triad which we have in this region here. So here we have a portal venule, a large structure, very little wall structure to it at all. Here we've got some bile duct tule here. In fact, we have two parts of the bile duct tule, so we've cut it just past a fork. There's this region here is bile duct tule, and this is bile duct tule, and then closely associated with the bile duct tule is some hepatic arteriole, and some of the other spaces here are lymphatic vessels. Really what we want to look at here is what looks like a thin inked line which is drawn here. Just inside, here's a plate of hepatocytes here. Why don't we go just to a little higher magnification. So here we have a plate of hepatocytes, and here we have the reticular fibers occupying the space of DISA and forming part of the sinusoidal lining. In a tissue preparation like this, we don't easily see the endothelial cells which line the sinusoids. Finally, to complete our study of the liver histologically, let's take a look at a small structure that's associated with the liver, is in fact adherent to the liver, and this is the gallbladder. And histologically it really couldn't be much simpler as organs go. The function of the gallbladder is to store and to concentrate bile. And as we look at this section through the wall of the gallbladder here, we can see that there's a mucosal or epithelial surface here. There's a submucosal area here, which is mainly muscle with some connective tissue, and then the outer coating of the gallbladder is a serosa or adventitial coat made of connective tissue with some mesothelium attached to it. So let's increase the magnification a little bit and take a look here at the mucosal coating. The surface is elaborated as a series of structures that look like these, almost like villi. I suppose perhaps it's questionable whether they really are villi, but it isn't really that important. Even at this magnification, you might be able to see that the epithelium is a simple columnar epithelium. These folds have a little core of lamina propria here. We can see some connective tissue in this region here, and then what looks like some smooth muscle here, some strips of smooth muscle interspersed with some connective tissue, and indeed this is the structure of the wall of the gallbladder. If we go a little bit higher, and again just wait for the slide to develop, and we'll take just a slightly closer look at the epithelium here. So the cells are sort of foamy looking, simple columnar epithelium, best seen along here. Here's the core of loose connective tissue, lamina propria, that occupies these folds here. There isn't really a muscularis mucosa. Some authors maintain there may be a 1 to 2 layer cell layer thick muscularis mucosa, but if there is, it's not easily seen here. So we have the epithelium sitting on its lamina propria forming the mucosa, and then we have a submucosal layer of connective tissue in which we have strips of longitudinally, as it is here, but circularly oriented around the axis of the gallbladder, longitudinally oriented smooth muscle, which we can see here. In this adventitial, or in this submucosal layer of connective tissue, in addition to the strips of smooth muscle, here's some cut in cross section, here's some in longitudinal section, we also get blood vessels. Here's an arteriole and a venule, and then outside this submucosal layer we see a layer of connective tissue. This is fairly dense irregular connective tissue which has torn apart quite a bit, and then finally a serosa or mesothelial layer which is adherent to the external surface.